Okay, thank you very much, ladies and gentlemen, and everyone for joining us today. We're going to go ahead and get started. We have a very tight agenda. A few housekeeping items. Number one is please mute your microphones if you're not going to be asking a question, so we don't get your background noise. And you're welcome to either leave your video on or turn it off during the presentations. And the other thing we want to mention to you is that we are recording, so the talks of our three outstanding speakers. So please keep that in mind. So with that, why don't we get started? My name is Kishore Wasan. I'm currently the co-director and co-founder of the Neglected Global Diseases Initiative. Most people, I think, on this call know who I am. I've got various degrees and backgrounds, so we won't spend time on me. And what we're going to do today is I'll do a little introduction of NGDI, turn over to my colleague and good friend, Dr. Richard Lester, to talk about the symposium. And then we have three sessions today with three outstanding speakers for your enjoyment and pleasure today. So as a background, and very short and sweet, in 2010, Dr. Bob Hancock and Bob has been an outstanding supporter of NGDI and helped me tremendously, and really this village of all of us to get NGDI going. We co-founded NGDI in 2010 with the support of many faculty and students and staff, and many of you who are on the call today. And the reason why we started NGDI is that we noticed that there were numerous faculty, students, staff, undergraduate, graduate students, postdocs, technicians, you name it, that were doing amazing neglected global disease research all across our UBC family and UBC campuses, but didn't really have a home and a place for where we could help facilitate their work as well as promote their work. And the question was, how could we help them and facilitate them? And really the byline that I heard over the years with NGDI was neglected folks doing neglected global disease research. And so that's why we decided to start NGDI almost over 10 years ago. And really the mission of NGDI has always been really the development of interventions and delivery of them to those of need. And with nothing else, I think the pandemic has emphasized the importance of this type of research. And today's symposium really highlights one of the really core principles of the Neglected Global Diseases Initiative, namely health equity. And so with that as a brief introduction, and you can learn more about NGDI by going to our website and following our Twitter feed, I'd like to turn this over now to my co-director, good friend and colleague, Dr. Richard Lester, who's going to introduce the symposium today. So over to you, Rich, and thank you everybody very much for being online today.
Thank you, Kish. So Dr. Wasan's understated self-accomplishments, I remember actually as I was new to UBC at the time, really, and coming from my work in East Africa and global health, really finding that a home where people were sharing ideas and really advocating for health equity, and NGDI has moved forward from there. And Kishan himself, I remember I was flying on an Air Canada flight when I saw the ad on my screen in front of me about his work on liposomal amphotericin B, and which has a huge potential in high pay markets, but also, of course, to treat maybe leishmania in remote areas because of delivery. So really, it's been an honor being part of this initiative. I'm thrilled today, really, about this symposium, which we've been talking about for several months now. We actually earlier in the year, and there was so much other going on that we debated timing, but I think it's very timely right now. We have some tremendous speakers, both UBC researchers, international on policy, and which I'll get into. I want to thank our organizing committee. So co-founders, as mentioned, Dr. Kish Wasan and Bob Hancock, who'll be moderating. Nancy Galini's previous Dean of Arts and economist. Dr. Catherine Pluis is doing global research in my division. By the way, I'm Rich Lester. I'm a physician researcher in the Division of Infectious Disease here at UBC. And Catherine's also in our division. Dr. David Patrick, who's does a lot of international work, important research lead in COVID in BC. Rachel Fernandez is our VP at UBC, has played a role in the organizing. And then we have our three distinguished speakers, which we'll be introducing as we go along. So just some quick background. People know the pandemic. We've now been living with it for almost 2 years globally. And you can see the number of global cases on this dashboard, this Johns Hopkins dashboard, at over 262 million cases. Vaccine delivery is now on these dashboards, which is a real win for science. And I think it really pulls into the role of science in treating pandemics. And we have eminent scientists on this panel. But equity is one of the things we're going to see. So you can see that this is the current pandemic. It's affecting almost every country to various degrees. And if I just move forward, if we look at Canada, again, this tends to come in waves. As you know, our very first wave last year, people were talking about a new normal a bit prematurely. Back in February then, that we might within a few weeks be back to a new normal. I think those with pandemic experience knew that we weren't out of the woods then, and we've had subsequent waves, and these all have a lot of impact, and not just deaths. Deaths are, of course, important to vulnerable people, but also societal things with lockdowns in the economy that really knock on. And I've got Rwanda here as an example, showing the different wave patterns in different regions of the world. And we have a eminent speaker, Dr. Sabin Nsanzimana, who's leading some of the COVID effort in that nation in East Africa. And they've had their biggest, most recent wave is finally down at the moment, but we don't know what's next. And as many of you know, there's new variants and lots of unknowns. So we'll hear from them. I had a quick look at what is the global research, because it's about science and access, and science has really been helpful. So if I did a quick PubMed research just on COVID, search on COVID-19, it came up with 202,000 papers have been published. And this is the word COVID-19 only came out in March, I think it was March of last year. So that's tremendous amount of published research. If you look at the WHO site, you can search on the COVID-19 database, and you get actually double that, almost 416,000 articles that you can. So there's a tremendous amount of research, but there's a lot of, is there global equity in that science and the benefit we're seeing? And I think we want to discuss that a bit today. Tremendous amount of research being done here at UBC. And you can go to the UBC sites to search many of our researchers who've had funded projects in various elements. And then also, but it really is about the world contributing. And we're very fortunate today. One of our speakers, Dr. Nsanzimana, from leading the Director General of the Rwanda Biomedical Center and leading their science of their COVID response. They actually had very early in the pandemic Nature article, which they wrote, lessons learned from Rwanda. And they did a lot of things better than many countries, in fact, in the speed of their response, data-driven and embracement of technology. And in fact, you can see an independent report done earlier this spring, updated, ranking government responses and how they've done in dealing with. And in the top, in fact, Rwanda got ranked, despite it being a lower middle income country, in the top 10 of its responses. Canada's, in the top half, but only at number 60. So I think there's lessons to learn and share all around. So global health equity is also about really sharing information, experience in all directions. Vaccinations is, of course, one of the scientific marvels of this pandemic. And but as we can see, delivery of that in Canada, we're at over like 90% of fully vaccinated, which is a great achievement relatively. But if you look at that globally, and this is a pandemic affecting almost every country, only 5.8% as of yesterday are thought in low-income countries have received at least one dose of the vaccine. And of course, that contributes to we're all in this together. We really need to think about that as we listen to our science and speakers today. And again, we're all part of this. The Sustainable Development Goals I'll point you to. Number 3 is really with the United Nations, which we all is about good health and well-being for all. And I think that fits into NGDI's founding principles, which is about trying to ensure equitable access to the science, technology, and policy that improves health worldwide. So I'm thrilled about our speakers. Others will be introducing them. So if you introduce yourselves and our speakers from there, just welcome and thank you very much. Looking forward to a interesting session, and there will be some question time at the end of each talk. Thank you.
All right. Well, thank you very much, Rich, for that wonderful introduction. Um, to get started in our first session, I will ask Dr. Bob Hancock, and everybody knows Bob. Bob is an outstanding microbiologist and world-renowned in his field, who will introduce our speaker and lead this first session. One thing I should, in remiss, NGDI is co-sponsoring this event with SkyMount Medical, and so I want to thank SkyMount Medical for providing some of the funds to help us put this on. So with that, over to you, Dr. Hancock.
Bob, you're on mute. Bob, you're on mute.
Okay, it helps if you actually get off mute. So, before I introduce Peter Cullis today, I'd like to briefly mention my own foray into COVID research. So we've developed a signature that enables physicians to predict severe sepsis and organ failure at first clinical presentation, namely in the emergency room. And this is important because in 2017, nearly 20% of all deaths on the planet were due to sepsis. And it's actually a major killer both in developed and developing countries. And this was likely even higher in 2020, since it's now known that patients dying of severe COVID disease succumb to, in fact, sepsis. Critical in these situations is to predict sepsis early on, since the timely initiation of therapy is key to increasing survival. So just by improving management protocols over the last decade or so, the death rate has decreased from 35% to, on average, 23%. So we've developed an early diagnostic gene expression signature that is 80% accurate in predicting severe sepsis, that is both regular sepsis and that caused in the context of severe COVID disease. And it does this within 1 to 2 hours of emergency room entry. And we're commercializing this through a company that I founded called Sepset, that as a result of financing is now majority owned by ASEP, that just went public on the Canadian Securities Exchange 10 days ago. Regarding developing countries, and this is really an important fact, we've been recently signed a term sheet with UBC to license in an additional sepsis technology from my lab. And we've defined in that term sheet a humanitarian objective where no profit is to be taken from lower income and lower middle income countries. So there's no question that COVID has made a huge impact on our lives, causing likely many more deaths than is evident from the official statistics. Where the impact is most underestimated is in poorer countries, lower income, lower middle income countries. But the actual impact is likely unknowable, since also access to vaccines is uneven, especially in these countries. And this uneven access is probably due to the logistics of delivery, to some extent to vaccine phobia in these developing countries, and to profound macroeconomic issues. So to set the scenes for our last two speakers who are going to address these types of issues, I'm really pleased to introduce a man who has literally changed the face of COVID through his tremendous discoveries. A PhD graduate in physics, Peter Cullis is a professor of biochemistry and molecular biology at UBC, and I'm proud to say I've known him since I first started at UBC and learned that he was a pioneer in the growing area of liposomes, now also called liposomal nanoparticles, and making numerous breakthrough observations. Peter recognized early on that this technology had the ability to concentrate and mask drugs to effect better delivery of those drugs. And he's since pioneered many first-in-class drugs, including liposomal amphotericin, an antifungal drug, liposomal doxorubicin, an anti-cancer drug, and liposomal small interfering RNAs for treatment of a hereditary disease called ATTR amyloidosis. This work has contributed to multiple drugs that have been approved by regulatory agencies in the US and Europe. Peter Cullis has co-founded more than 10, I think it's 12 biotech companies, and has established published more than 300 to 400 articles, is an inventor of over 60 patents, co-founded the BC Personalized Medicine Initiative and the Center for Drug Research and Development. And he's received many awards and is a fellow of the Royal Society of Canada, and also was awarded the Prix Galien, which is the Canada's premier prize for achievements in pharmaceutical R&D. Recently, he was the father of the Pfizer-BioNTech COVID-19 mRNA vaccine that absolutely relies on liposomes for its effectiveness. This has led to more than 250 million doses of this vaccine applied in the USA alone and billions of doses worldwide. For his research, he recently won the prestigious 2021 Prince Mahidol Award for Medicine, which is out of Thailand. In the Globe and Mail, when they reported this award to Peter, they asked him what it was like as a scientist when he went for his own first vaccine shot. You know, he told the nurse who was administering the jab, "I actually had something to do with making this vaccine." She responded by shooting him a glance that was more odd than awed. Like I was from outer space, Peter Cullis recalled. So joining us from outer space, I'd like to introduce you today, Peter Cullis, who's going to talk about what you can see on your screen. Thanks, Peter.
Yeah, thanks very much, Bob, for that introduction. Yeah, it was. She did look at me like, man, oh man, I mean, this cannot be true. So I didn't pursue the conversation that much. But what I'm going to describe today is this title here is a 40-year journey to the. I think I have about 30 minutes to do this, so I've got too many slides, so I'll have to go through some of them quite quickly. But it's a 40-year journey. What I want to do in this is to emphasize the importance of basic research to some of these translational activities that Bob described. So the alternative title is Lipid Nanoparticles that Enable Gene Therapies. I have a number of conflicts of interest, Precision Nanosystems and Acuitas, where I'm a co-founder, and Nanovation, where I am a co-founder and chairman. Okay, so what you often see in the popular literature describing the vaccine, the mRNA vaccines, is that they're coming, as is termed here, inside tiny bubbles of fat. And this is the delivery system that we played a major role in, as I'll describe. Now, just to be a bit more clear on these tiny bubbles of fat, what are they? Well, they're pretty tiny, 100 nanometers or smaller. We usually work in the 60 to 80 nanometer range, but they're really not bubbles of fat, at least not fat as you normally think about it. They're nanoparticles that are made of membrane lipids, and they're product of a story that for me started over 40 years ago. So I got my PhD way back, and this is in solid state physics in 1972. And I realized that I wanted to do something different outside the field of physics, and I got interested in biological membranes. Now, what do you learn as a physicist? I mean, you learn a number of things actually. One of them is you learn a technique. So I knew a bit about NMR, nuclear magnetic resonance. As a physicist, you get taught to build things if they're not available. But the basic thing is to go after fundamental problems. And so when you're talking about biological membranes, what were the fundamental problems in those days? There's still problems actually that remain today. But on the left, you'll see the question we were asking at that point. Well, why are biological membranes contain a lot of lipids that don't like a bilayer structure? I mean, obviously the biological membrane as indicated here relies on a bilayer structure of the lipids to maintain a permeability barrier. Why do you have all these lipids in there that adopt, if you extract them and put them in water, they adopt these non-bilayer structures, hexagonal H2 phase being one of them. And the other problem we got interested in was that biological membranes have asymmetric transbilayer distributions of lipids. In other words, that the lipids on one side have a different composition than the lipids on the other. So we said, can we drive this using the ion gradients that are across membranes, and what are the consequences? So these are about as far away from developing a vaccine as one can imagine. But in the end, these questions did lead to that result. So we used phosphorus NMR to study lipid polymorphism, and you'll see this this in a later slide. If you have a phospholipid, has a phosphorus atom obviously in the head group. If it's in a bilayer, you see one line shape. If it's in hexagonal phase, you'll see another line shape. If it's in other structures, then you see different line shapes. And so you can use phosphorus NMR as a signature of different structures. You can also generate lipid asymmetry using model membrane systems. And in this case, what we did was we synthesized what's we termed an ionizable cationic lipid called DODAP. It can exist in a protonated form or non-protonated form. And we could generate lipid asymmetry because if this lipid was present in a say 5% in a host bilayer, it will preferentially distribute to the side that's the most acid, where it gets protonated and can't get back out. And so we could drive lipid asymmetry in that manner. And that's the basic work. And I'm not going to go more into it, but that led to lipid nanoparticles that enable the vaccine and other gene therapies. So here I'm going to talk about three topics: packaging nucleic acid polymers. This is work that's gone on for the last 25 years. Work that we did between 2005 and 2012 on siRNA, small interfering RNA, and then subsequently a few slides on the vaccine application. So the challenge that we took on in 1995 was to develop a delivery system to take nucleic acid-based drugs to the liver and enable intracellular delivery into hepatocytes. This is a tall order, as anybody that knows anything about the immune system would acknowledge. We're taking genetic material, we're injecting it IV, it has to circulate around, say, get to the liver, extravasate, get taken up by hepatocytes, and then not only that, but break out of an endosome to deliver the contents to the inside of a target cell, to the cytoplasm. So we hit a first problem right off the bat. I mean, how could we encapsulate nucleic acid polymers in these lipid nanoparticles? We require positively charged lipids, obviously enough. This is where we can get a hydrophobic complex. However, there's no cationic lipids in nature. They're extremely toxic molecules. There's only net neutral lipids or negatively charged lipids. And so we had to be able to use a cationic lipid, but have one that's not toxic. And this is where our basic research came in quite handy. Because we went back to the ionizable cation lipids we devised for lipid asymmetry to determine whether or not we could use them as ways to encapsulate nucleic acid polymers. So they're protonated and positively charged at, say, pH 4, about the acidity of a lemon, whereas at neutral pH, they're deprotonated and neutral. So what we found was that we could use these lipids to load the negatively charged polymers into lipid nanoparticles at pH 4. And then when we raised the system to pH 7, they were retained, much less toxic. And they also turned out, as I'll indicate, to have important properties for intracellular delivery. So this is the first ionizable lipid, just to reiterate again, pH 4, they're protonated at pH 7, net neutral. And what we found also was, okay, how do we encapsulate? Is there a process we can use to encapsulate these nucleic acid polymers? And we developed a rapid mixing ethanol dilution process. So where the lipids are dissolved in ethanol, the oligo that we want to encapsulate is dissolved in water at pH 4. Mix these rapidly, and intuitively you can see that the first thing to fall out of solution here is going to be, say, the oligo with the positively charged lipid around it. If we do this fast enough, we can trap it in a limit size state, say the PEG lipids that will come out of solution and stabilize these nanoparticles. So these are a new class of nanoparticles with a hydrophobic core, no limit to the size that we can encapsulate, 100% efficiency. We can dial the size by changing the surface lipid to core lipid ratio, relatively non-toxic, scalable, reproducible, etc. And so this was the first step in this process. So let's go on now to the Onpattro story, as it's termed, which is developing an siRNA-based drug to treat a condition known as hereditary amyloid transthyretin-induced amyloidosis. So our objective in this period was to silence genes in the liver, develop an siRNA lipid nanoparticle siRNA system to silence genes in the liver. Obviously, many diseases come into focus if we can silence or express or edit a gene in the liver. This is just a small subset of those disorders. And we started with the question that can these lipid nanoparticle systems containing the ionizable lipids silence genes in hepatocytes? And this was using a factor 7 model. So we had LNPs that are containing siRNA to silence factor 7, the clotting protein. So this would then involve injecting this by the tail vein at time 0, terminating the mice and assaying the plasma for factor 7, say 24 hours later. And this was a fairly high throughput model, so we could run 2 experiments a week in our efforts to optimize these systems. Now, just to guide ourselves, in terms of how we might design these systems, LNPs are taken up by endocytosis, and what we basically want to do is break out of the endosome before they get to the lysosome, where of course everything is going to get chewed up. And so we design these systems so they break out at a vulnerable stage. Now, how can ionizable lipids destabilize endosomes? Now, what we found was that this were basic studies again, that cationic lipids combined with endogenous negatively charged lipids to induce non-binary structures such as the hexagonal H2 phase. This is a representative phosphorus NMR spectra here. We have, say, phosphatidylserine, an anionic lipid, add a cationic lipid to it, and it flips over to these non-binary structures. Be rationalized as a change in the shape of this complex, which favors this structure versus a bilayer structure. And so what we said, okay, well, we need to design ionizable lipids with optimized shape and pKa properties, going back to lipid polymorphism. Again, just showing how one relies on basic science studies or basic properties of lipids to design these systems. Maximum cone shape, as we might term it, small head group, large acyl chain area, and then so that they get protonated at the right point, and optimized pKa. Now, we found that the potency of these systems was really highly sensitive to the pH at which the ionizable lipid becomes protonated. Synthesized over 300 lipids. This ionizable lipids. This was work done with Alnylam, a company based in Boston, and and moved through a series of more and more active lipid nanoparticle systems in terms of being able to silence genes, to silence the factor 7 gene in hepatocytes. So here's a graph, the log plot on the y-axis of the potency of these systems in terms of being able to silence these things. This is 1 over the ED50, where the ED50 is the effective dose to get 50% gene silencing of factor 7 in the liver. And as you can see, what we identified was a marked dependence on the point at which these lipids get protonated. Those lipids which have a pKa of approximately 6.4, if you move as little as half a unit away, the potency could go down by 2 orders of magnitude. And so this was really a seminal discovery. So obviously we didn't expect this from the beginning with our introduction of these ionizable lipids, but they've proven to be markedly useful. So this indicates where we got to over a 5-year, 6-year period. We moved from systems where the dose needed was 10 mg per kilogram of siRNA, down to 5 micrograms of siRNA per kilogram body weight in these mouse models. And so at this point, we said, okay, well, let's move this into the clinic. And the clinicians at Alnylam chose this disorder, the hereditary amyloid transthyretin amyloidosis. This is a multisystemic disorder. That's obviously a genetic disorder, but there's many mutations in the transthyretin gene, which of course made in hepatocytes, that can cause the formation of fibrils, which deposit pretty much everywhere, but have nasty side effects in nervous tissue and cardiac tissue. So neuropathies and cardiac problems resulting from that. Really no effective therapy, and it's usually fatal within 5 years of a diagnosis. So a pretty nasty disease is indicated here. And obviously, for individuals, they've often got the first indication of difficulty walking when they're about 30. They would know what they're in for because they would have seen a relative die of this disorder. So this is where gene therapies are very attractive, because the hypothesis you can come up with is very simple. In this case, what we're saying is, okay, well, let's silence the transthyretin that's made in the liver, going to reduce the formation of these fibrils. Maybe if we're really lucky, we'll get a stabilization and recovery if we can dissolve some of the already formed plaques. So a potentially simple solution to a devastating disease. So this was the clinical development program that was put in place by Alnylam starting in about March of 2012, when the clinical trial results were announced in 2017. First in healthy volunteers, and then subsequently in a phase 3 trial. So this was the results in healthy volunteers. That's at approximately 0.15 milligrams per kilogram in these individuals. You see this pronounced and sustained silencing of transthyretin in the circulation as a result of the IV injection of lipid nanoparticles containing siRNA to silence that particular gene. And so this, as a result, this was selected a dose of 0.3 mg of siRNA per kg body weight every 3 weeks. You can see that the silencing is maintained over 3 weeks for subsequent clinical trials. Then moved into a phase 3 trial as an 18-month phase 3 study. And the 225 patients, double blinded, et cetera, 148 in the treatment arm, and 77 getting sterile saline, which was the placebo. Primary endpoints being change in neural impairment score over this 18-month period. So a number of other secondary endpoints that I'll go into in a minute. This is astonishing results that were observed here over this 18-month period, because whereas those on the sterile saline got steadily worse, their neural impairment score got higher. Those that were being treated with the transthyretin siRNA to silence transthyretin, if anything, got better. And so we're reversing the effects of an hereditary disorder, as you can see here. So this was the phase 3 trial results that were announced in 2017. Hit the primary endpoint, which was the neural impairment score, and every secondary endpoint as well, with p-values that are just, I don't know why they bothered to calculate them. This drug clearly works. The quality of life and muscle strength, nutritional status, etc., all demonstrating very, very pronounced benefit. So patisiran, as the drug was called then, it's on Patro now, is a stabilizing, possibly curative therapy for a previously fatal disorder. So this was approved by the FDA in 2018, the first FDA approval of an siRNA-based gene therapy drug. So a pretty big deal, not only being able to halt the progression of hereditary disease, but reversing accumulated damage. And as I say, really does demonstrate the power of these gene therapy approaches. Okay, so now I'm just going to go on and close with the BNT162b2 story, which is the COVID-19 story, which took place over the last 8 years, and has largely been conducted at Acuitas, one of the companies that I co-founded in 2009. So the here we ask the question, does the lipid nanoparticle technology we developed for siRNA, does that also enable mRNA-based gene therapies? So here, instead of packaging the siRNA, we're packaging the messenger RNA and lipid nanoparticle and going through the same process. If we can deliver the siRNA to the cytoplasm of hepatocytes, then maybe we can deliver messenger RNA as well and get gene expression. It turned out that the rapid mixing ethanol dilution procedures employed for siRNA were equally applicable to messenger RNA. Of course, whereas we might have 700 copies of siRNA per nanoparticle, here we might only have 3 or 4, with this much larger messenger RNA. But from a morphological point of view, these things look very similar. These lipid nanoparticles look very similar to what we would see for the sRNA containing agents. The bar here is 100 nanometer bar. This is just indicating for luciferase, lipid nanoparticles containing luciferase mRNA. Some initial experiments just indicating that the liver will light up in a dose proportional manner. So indicating right off the bat that we are getting gene expression in the liver as a result of administering these lipid nanoparticles containing this message. Of course, this means basically we can express any protein we want. And Acuitas, that's the major emphasis of Acuitas is developing better and better systems for getting gene expression in the liver. This is a for non-human primates indicating the present stage where developed or administering lipid nanoparticles containing mRNA coding for the heavy and light chains of an antibody, of a monoclonal. And you can see we're getting very high levels in the circulation with sustained circulation lifetimes. And obviously a huge number of applications that can come from this, where we can express essentially any protein we want, not only antibodies, but proteins that perhaps are not being made as a result of some genetic deficiency. But I'm not going to talk about that. Let's talk about more about serendipity, because in 2014, we were approached by Drew Weissman, who'd been working with Catalin Carrico for many years, to explore the potential of mRNA therapeutics as vaccines. And they'd found a way of modifying mRNA to reduce immunogenicity and increase gene expression. But Drew said, we got a delivery problem. How do we get our mRNA coding for these viral proteins into muscle and immune cells in order to make an effective vaccine? And so this just indicates what was needed. Coding for the protein associated with a virus, getting that made in either muscle cells or in antigen presenting cells, and getting MHC1, MHC2 class responses to the synthesized viral protein. And so this was work that was conducted over is still going on actually, conducted over 6 years, mainly based on influenza, but I'll just show and just indicate very briefly results for Zika virus, where the mRNA is now coding for the pre-membrane and envelope glycoprotein of the Zika virus. And this was published in 2017. And the results showed here that there was complete protection against Zika virus, as is injecting lipid nanoparticles containing mRNA coding for the surface protein, and injecting that intradermally, and then challenging at 2 weeks or 20 weeks. So I'm not going to show the data there just in the interest of time, but there was really total protection as a result. But what occurred here was that Acuitas, as a result of the Zika virus and influenza work and others, Acuitas has started working with BioNTech to develop influenza vaccines, and BioNTech was also working with Pfizer. And so when the pandemic hit in January 2020, all efforts switched to making a COVID-19 vaccine, where of course the messenger RNA now is coding for the SARS-CoV-2 spike glycoprotein. And so this was as everybody is aware, a rather successful venture that was the results of which were announced in November of 2020, demonstrating that this vaccine was 95% effective against COVID-19, irrespective of age or gender and race, and it was reasonably well tolerated across all populations. I should say 43,000 participants, probably one of the largest trials conducted in the area of vaccines. And so this is older statements here. expect to produce globally 50 million vaccine doses, 1.3 by the end of 2021. I think Pfizer BioNTech is delivering 3 billion doses by the end of this year and now producing at the rate of 500 million doses per week. So the lipid nanoparticle mRNA systems are really playing a major role in ending the COVID-19 pandemic, and has been approved in numerous jurisdictions, as we're all aware. But really, the vaccine applications are just the tip of the iceberg. Obviously, there's a lot of other vaccines that are coming into focus here and that are being developed as we speak. Universal influenza vaccine. The results here look very very encouraging. HIV, again, very encouraging. Zika, as I indicated, malaria, the list goes on. Cancer vaccines, and of course you can produce antibodies against the cancer, heart disease. So there's various, a result that came out a little while ago is an effective vaccine against atherosclerosis by knocking down LDL levels. Alzheimer's, inherited diseases, rare diseases, again, you can see the way forward here. So that's the story that I've been involved in over the last 40 years. So first of all, packaging. Secondly, the sRNA story for Onpattro, and then third, how we were developing something going for one direction, and then a collaboration with Drew Weissman et al. Really changed that and to develop a COVID-19 vaccine. So it's been a pretty long journey. But what I want to emphasize, just to close, is it all arose from very basic research that had no therapeutic intent at all. This: why are lipids that adopt these non-bilayer structures present in membranes? And can we generate lipid asymmetry using ion gradients? And in particular, what we were focused on were proton gradients. And so this is I think, a lesson that things are not linear in science, and there's no linear path to therapeutics we might want in the future. Just want to acknowledge a few people here. Particularly Mick Hope and Tom Madden, who I've worked with for 40 years. They were postdocs in my lab back in 1981 and 82. And as well as Steve Ansel, Ying Tam, Barb Wee, Paul Lin, I worked with for over 20 years. And we've had these very long-term relationships, and that's what's really enabled this whole process. People at Alnylam who were totally instrumental in making this go forward: Mark Tracy, Kin, Martin, and Mano Manoharan. My own lab, and of course Drew Weissman at the University of Pennsylvania. And so with that, I will close and take any questions that people may have.
Thanks, Peter. We have 10 minutes for questions. So I'm going to invite members of the audience to either raise their hand, which is just under reactions at the bottom of your screen, or to put a question into the chat box. So I'm just going to start off. Peter, I just want to ask about two things. So firstly, how cost effective is this? Because this obviously relates to how well it's going to be deliverable in a developing country. And is the Moderna vaccine the same basic type of technology?
No, I'll take your last question first. Yeah, it's basically the same technology. I mean, Moderna was following what we were doing with the sRNA, with the Onpattro drug, and essentially developed their own version. In terms of the cost effectiveness, there's two parts to this. I mean, one is the very rapid way in which you can generate these systems. As you saw, they were once you know the sequence of the virus, then you can have a vaccine ready to go in the clinic within 2 or 3 months, and that's counting doing a fair amount of preclinical work. So it's very, very fast. And there's a number of obstacles that are present now. One of them is this minus 80 cold chain requirement, which is an issue. There's no intrinsic reason why that needs to be, and really it was hard to do a year stability study when they had to get something in the clinic within a couple of months. I'm sure that those problems are going to be overcome. The intrinsic costs here are not huge, so it is the manufacturing, of course, is much more straightforward because you don't need cell-based approaches to make this vaccine. So it's I think from a developing country point of view, that setting up a manufacturing suite that can manufacture these things at scale is a real possibility.
So I have to ask, because you opened up the possibility of these questions. HIV and measles, and malaria, two massive issues where the vaccines have been a little bit indifferent, like 50% efficacy. Are you getting better efficacy with the mRNA vaccines? Do you know?
It would appear so. Moderna just announced some results that looked quite promising. And then I know that Acuitas has been collaborating with Barton Haynes and other people at Duke on an NIH-based approach, which is also looking promising for HIV. But I'm not that familiar with those results.
So Sultan Muhammad has asked the question, could adding an appropriate adjuvant be the potential way to increase the durability of protection for current mRNA vaccines? In other words, can you further improve on this technology?
I have to preface my remarks by saying I'm not an immunologist, but one of the things that's come out of this, and Drew Weissman has been working in this area quite extensively, is that the ionizable lipid seems to have a pronounced adjuvant effect itself that is actually very beneficial. This production of the T follicular helper cells in the lymph nodes that results in a large part of the potency of these type of vaccines. Whether that can be improved, I don't know, but I would suspect it could be. Obviously, these things were designed to get better expression in the liver and were more or less transferred wholesale to a vaccine application without much work being done in the middle. I think people are now hard at work saying, well, how do we optimize these systems further for this particular vaccine application?
Okay, Dave Patrick has a question.
Peter, thanks very much. So the Omicron situation has been a bit of an indictment of our ability to deliver vaccine equitably globally, obviously. I'm watching what's happened with genomics, where a technological breakthrough like Oxford Nanopore has sort of democratized and globalized sequencing capacity. And in fact, South Africa just paid the price for having taken up that technology, unfortunately. But I'm wondering if you can flesh out a little more. You mentioned in your reply to Bob that you could see scaling up manufacturing elsewhere. Can you paint us a vision of what a more globally distributed mRNA vaccine production and distribution capacity could look like? Have you thought about that?
Oh, yeah, definitely. And obviously, I mean, we're not a third world country, but Canada is equally bereft of the ability to make vaccines, which is a stupid situation. Anyway, in terms, one of the things that to realize here is that the quantities of material are not huge. I mean, the Pfizer BioNTech vaccine uses 30 micrograms of mRNA per dose, which means a couple of teaspoons is good enough for 1 million people. So the mRNA production is not a huge issue. The lipids are relatively straightforward to synthesize. I should say that that's the biggest holdup right now in terms of production. Is the raw materials. The FDA likes cholesterol, for example, and there's a component of cholesterol in the systems to be synthetic cholesterol and not derived from animal sources. I think so these are the synthesis of some of these lipids does limit production. The real basic problem here is that companies, I mean, for third world countries, is that companies like Pfizer, which can take to build a greenfield operation is going to take you a couple of years. For the companies like Pfizer are just taking over other manufacturing lines for other drugs and saying, okay, sorry guys, we're now going to make vaccines here. And they'll be at 5 billion, probably more than that, production next year. And producing it very, very effectively. So the possibilities are here of building things which could become white elephants. You know, in other words, you're producing material for a problem that's gone by. So it's a difficult decision as to how exactly to democratize this type of technology.
Okay, Abhishek Wadhwa has asked, firstly, he thanks you for your wonderful talk, which I also would like to thank you for. So this is the last question. So what are your thoughts about, instead of using messenger RNA, using delivering CRISPR-Cas9 that would then allow you to address the challenge of repeatedly using siRNA liposomal nanoparticle administration?
Oh yeah, no, this is already happening. For the, I mean, for example, for the transthyretin-induced amyloidosis, we can take siRNA to silence that, and as you say, give it every 3 weeks. But the company called Intellia has just brought out a CRISPR approach where they're nicking the transthyretin gene. And yeah, it's but one shot, this one and done, one shot and you're finished. So these approaches are, you know, the mRNA approaches are really quite revolutionary in terms of their potential.
Okay, thank you, Peter. That was a brilliant talk, and I'd like to get everybody to join me in thanking Peter. You can do this under the reactions button. You can clap like I am here.
Peter, yeah, this is Keish. Thank you so much. Really appreciate it. I know you've been a very busy man. One thing I will like to mention is you have been a pioneer for drug delivery in Canada, and thank you for the Nanomedicine Innovation Network. I think you've helped a lot of us in the area of drug delivery and nanomedicine, and a pioneer for the next generation of scientists in this area. And I think I've told you that personally, but thank you, Peter, for all that. Plus, my wife told me to tell you that. So for many that don't know, Peter was on my wife's PhD committee, so I thought I'd better say that.
We do go way back.
That's right. So thank you so much, Peter. And
Yeah,
thank you,
thanks,
Bob.
Chris.
You bet. All right. So let's move to our next session. And I'd like to ask Dr. Catherine Pluis to introduce our next speaker. So Catherine, over to you.
Thanks so much, Kish. Hi, everyone. I'm Dr. Catherine Pluis, as Rich kindly introduced. I'm part of the Division of Adult Infectious Disease at UBC, and I'm honored to be a part of NGDI as part of a board member, and a member also as part of my own research internationally. But I'm very excited to introduce our second speaker as part of the panel 2, Access and Delivery. And that's Dr. Sabin Sanznimana, and he's a physician and an epidemiologist currently serving as the Director General of Rwanda Biomedical Center, which is the implementing entity of Rwanda Ministry of Health. He is the leading COVID-19 response, or he's leading the COVID-19 response, and has led the HIV and hepatitis division at the Rwanda Biomedical Center for over 9 years, with an extensive experience in the HIV program design, strategic planning, implementation, operational research, and with a focus on global care and treatment of people living with HIV. Dr. Sabin served as principal investigator for several large research projects, including clinical trials in Rwanda and a multi-country research collaboration. He has served on several HIV guideline development group panels of the WHO, and he's also a peer reviewer for scientific journals and has published extensively on HIV, STI, tuberculosis, viral hepatitis, and cancer in Rwanda and globally. Dr. Sabin is a fellow at the African Scientific Institute and serves as adjunct assistant professor of global health delivery at the University of Global Health Equity in Kigali, Rwanda. So I welcome Dr. Zaznimana to take over the stage. Thanks very much for joining us.
Thank you very much for having me. I hope you can hear me, and it's a pleasure. I'm joining from Kigali, Rwanda, and I've been following the previous session. It's quite interesting. So I'm actually going.
We're having a little bit of trouble hearing your audio, Dr. Zaznimana. I'm not sure.
Yeah, I will increase my voice if that is good. Just let me know.
That's perfect.
Thank you. Yeah, you sound, this is Kish Masan, you sound great right now. So if you could keep your voice there, that'd be perfect. Can you share your screen?
Sure, I'll be trying to bring on my screen. You take maybe a few seconds, but in the meantime, I can move on, because I'll be talking about. I'll be speaking about what is already on the slides if they don't happen to appear. So moving on, I was going to tell you that when COVID came to Rwanda, no one really knew that it was going to last so long. And the response and the teams that we put in place in the country did not take into consideration that over 2 years we'll still be fighting this virus that was not even much known in the context that it has affected all of us. So the first case confirmed in Rwanda in 2020 in March was kind of a shock and a panic across the whole country, given that what we've been seeing and observing in the media across the world, coming from one country reaching so many countries. And it started as one case, but very rapidly it has been affecting almost all parts of the country, including community transmissions or across the country. And the first wave that we really experienced was quite less severe than the second one that came towards August, 6 months, 5-6 months later. And then in December 2020, beginning this year, that's where the country was severely hit by a second wave towards June, July, when more cases actually increased, and in a very abrupt way when the neighboring city of Goma in Democratic Republic of Congo was affected by volcanic eruptions, and the entire city of Goma, people moved into Rwanda in just one night. And you can imagine the cross contamination, mobility of people that happened. And actually that is how the Delta new wave started in our context in Rwanda, and later on has become a dominant variant across the world until now. So we've been learning and experiencing a lot, putting in place mechanism to fight COVID using different technologies that were actually being brought throughout this pandemic. First, communicating to the people. When people were actually into total lockdown at home, it was very hard to reach to them, because usual ways people are gathering in places, then are given messages. Imagine if we're staying at home, the only ways were actually to fly. I mean, to go to the household and tell what was expecting them, bringing their own mask when everything is getting better a bit outside, or even helping those who are in home-based care. So we started by using drones, passing messages across communities, and in the treatment centers also. We've been trying ways to limit the contaminations from patient to doctors with limited protection that in the beginning were actually very hard to find personal protective equipment. But we've been able to acquire some robots that we deployed in all treatment centers. They were basically helping to keep medical records and taking vital signs like temperature, like serving food and drinks without many contacts of healthcare workers or cleaners going into the patient rooms in regular ways, exposing them at different times. So using robots and drones have been the first, I would say, experience of trying the technology in our context. But later on, we were necessary as we get more data from testing, from case management on how we were going to keep these informations and how people tested, received their result without coming back to the lab. So we designed different systems first for the lab, so that we can give results using text or using emails or using other technology ways of getting results. And for home-based care, we were glad to partner with Welltel company. My colleagues and friends, Dr. Rich and his team, were able to actually very early days of COVID response, we quickly adjusted this Welltel system that we had used before on HIV to also adapt it to COVID-19 home-based care. So that system was helpful because you could record everyone who was tested positive, and whenever they're at home, you receive SMS in both directions. Then they can also check on the daily status and exchange with a team of healthcare workers or rapid response teams without going into people's households. So the Welltel system has been quite helpful and is still used even now in Rwanda in all the COVID response systems. It was a good learning process from testing, from home-based care, and also vaccination. When it started in March this year, it was a quick design of digital tools that will provide information to whoever is coming to vaccine, receive a vaccine. Whenever you receive vaccines, is SMS generated, and you receive a code that helps to download the certificate immediately. That was actually last year, and until now, we've vaccinated for at least for first dose more than 6 million people in Rwanda, the country of 12 million people. And for second dose, we've covered more than 4 million. So it's a process that has gone up quickly. But today, I want just to highlight the importance of digital tools that would help from screening, testing, diagnostics, treatment, vaccination, and even sharing data in real time that has helped also to update the populations every evening. The systems, everyone testing in Rwanda, data is flowing in real time to our digital tools, and we could also put that to the every evening report, being in treatment centers, and that has been a learning process, adapting to what we've been receiving and seeing every day. And today, as now we're facing a new wave, probably coming soon or not. You've talked about the new variants and how the sequencing capacities are being improved in our lab. We are sequencing samples every week. And so far, although the Omicron new variant is not yet detected here, but alert system is now at maximum levels, so that whenever we detect it, we do contact tracing, and using these tools, we can know also who has been in contact with which person, to be able to do testing. So to cut the story short, so maybe interaction questions will be much easier. I want just to highlight that with COVID, we've learned a few things, but very important that we can do things fast and we can innovate much more under this pandemic, the pressure. Number two is the partnership that we've acquired both on the continent, but also beyond. As I mentioned, with several initiatives and ideas. And number three is that the scientific developments that we see and what we know and what we don't know is actually growing more and more. We are being challenged every day, and the challenges are helping us to even think further. To end, I want just to highlight that the Rwanda is looking now into vaccine manufacturing. We've started initial agreement and steps with biotech company on vaccines candidates that may be helpful to tackle different infectious diseases. This has been an exciting moment, both as we think of a continent that has been actually importing 99% of the vaccine needs, being able to think of how we can now produce vaccines on the continent. If you look at the map of countries that are vaccinated or already covered, Africa is lagging behind less than 10%, and that is the biggest challenge. If you look at how we can tackle a disease in a global context, these differences must be addressed. That's how we can end and actually control in a sustainable way this pandemic. So I would like to pause here and thank you again for invitation and for everyone who is on this panel and this session. Thank you so much, Chair. Over to you.
Thanks very much, Dr. Sanzimana, for that very interesting perspective and the use of this, how you've applied these amazing technologies with respect to the COVID pandemic. I just want to open up for the audience if there's any questions.
Hi, Catherine. It's Keish. Sorry, I took off my video. So my question is, what's happening in Rwanda in terms of variants, the Delta variant, the Delta Plus variant, and how is the Rwanda government trying to address the variant situation?
Thank you very much. That's a good question, I think, applied to any other country in the world currently. So what you're doing first is to ensure that the surveillance systems and testing are working from the entry points, especially at the airports. So everyone arriving at Kigali International Airport, for example, must present a negative PCR test result before even departure. And at arrival, we also do another PCR test at the airport. We've been able to establish a PCR lab within the airport so that we cut the turnaround time that provides result within 4 to 5 hours. This helps detect any positive case being COVID, any type of variant, and also do a necessary contact tracing. Besides that, within our lab, we've been upgrading our sequencing capacities. So we are doing sequencing on a regular basis. So far, we haven't seen any Omicron case, but Delta remains the dominant variant. Actually, this week sequences are showing 100% of the samples were Delta in Rwanda. So we're doing it at different points at the borders, because we share border with 4 countries, and at each border we have these testing systems. It's quite a lot of work of the teams that are working days, nights, but it's helpful because whenever we see a surge of cases, it's easy to detect and respond on time. Over.
Great.
Thank you.
All right. Thank you very much. Love that. Thank you. Appreciate it.
I just had another question. It was impressive. You mentioned in terms of second dose of vaccinations, I think you mentioned over about 4 million people have already had their second dose, which is phenomenal. I just had a question related to kind of cold chain. I have my own interests and work that I do within cold chain, more related to sort of ice-less based technologies for vaccine delivery. What sort of technology is being used in Rwanda for the actual vaccine delivery and cold chain?
So thank you very much. For vaccines, I think we first, before even the vaccine, the COVID vaccines were developed, we've been having our capacities improved in terms of preparing for a vaccine that will require deep freezer storage system. So we acquired deep freezers early in December last year, because we've been following all these developments in the mRNA vaccine development. So Rwanda actually was the first country in Africa to use Pfizer vaccines, because at that time we had built this capacity. And up to now, this is the vaccine we have used the most, representing actually half of all vaccines we used. Followed by other vaccines like AstraZeneca, that's quite easier to store, +2+8. And we've used also Moderna vaccines and Johnson & Johnson vaccines. And the way these vaccines were deployed were generally looking at categories of categories of people we've been first addressing, like healthcare workers, people with comorbidities, elders, and also targeting people different age groups. So today, the eligibility criteria now is 12 years old and above, receiving vaccines. And we have started since this, actually last week, to give a third dose for those who received 2-dose regime vaccine, and the second dose for Johnson, those who received Johnson vaccine. So this is how our program is evolving, vaccination. Unfortunately, we don't have enough vaccines for everyone in need, and we don't have actually issue of vaccine storage or supply chain or vaccine hesitancy. These are issues that probably I would say we've heard about in some places, but not in Rwanda.
Excellent.
I have a question. Thank you so much for your talk. My name is Nancy Galini, and it's just wonderful to hear the tremendous achievements being made in Rwanda. But my question involves you mentioned that Rwanda is looking towards manufacturing vaccines, and I'm wondering what kind of cooperation you're getting from pharmaceutical companies on that? Are they agreeing to not assert their intellectual property rights? Are they licensing local manufacturers? Could you talk a little bit more about that?
Yes, indeed. That's a very good question. We've started this discussion with vaccine manufacturing with different companies, but so far the advanced the discussion and talks with biotech company involving also not only Rwanda, but also other African countries like Senegal and potentially other hubs. We have 3 hubs of vaccine in Africa, Rwanda, Senegal, and South Africa. But the way we're discussing it and how we see it is working together with BioNTech on establishing these facilities in our countries, and working hand in hand, and also having necessary certification and approvals that will later on be handed over to the to those countries as skills and capacity are transferred through a certain period that will be agreed upon. It's still at early stages, but some necessary steps have been made and a lot of announcement already done and agreement signed. So we look forward to that, and it's expected that by next year or mid next year, this will be moving to implementation and make it happen.
Thanks so much. It sounds so promising.
Thank you.
So I'd like to ask a question, please. So I've read in South Africa, the compliance with vaccination is extremely low, I think in the low 40%. And it sounds like in Rwanda, you're doing a lot better. And I wonder, how are you encouraging citizens that vaccines are a good thing, as opposed to a stab in the arm with a metal object?
Thank you. I mean, before we start vaccination of COVID-19, we did a survey. Rwanda Biomedical Center conducted a survey. We asked thousands of people if COVID-19 vaccine is coming, who was willing to take it. And we were actually surprised to see that 86% of people responded that they would be willing to take the vaccine if it was approved by usual bodies or institutions that have been bringing vaccines. Because for other vaccines, the uptake has been always above 95%. And we received almost exactly the same kind of feedback, because people trust in vaccines has been a culture built over before, and COVID-19 vaccine came in as just vaccine number 15 out of other 14 vaccines we've been providing. It wasn't an issue. The beginning, we were quite surprised because reading in the news and what was being discussed in other countries was quite scaring and a concern. But I would say the vaccine acceptance or uptake won't be built during the distribution of vaccines. It's something that has to start earlier, before, many years before, and that trust has really helped us to prepare the ground for a COVID vaccine. Up to now, the uptake and people really coming forward to take the vaccine is above 90%, far beyond the 86 we had surveyed before in February last year. So we still have a few groups here and there, especially related to beliefs and convictions that are associated to small religious congregations that but represent really a very, very minimal categories of people that the only challenge I would say we have, especially in the rural areas. Thank you.
Maybe Hello, can I?
Is there, yeah, any other questions? I think Rich, yeah, go ahead.
My real hand as I'm struggling to find the button. Thank you, and Dr. Zaman. In terms of so we heard from Dr. Peter Cullis, the amazing story and the long story of science that really led to some of the technology in these vaccines, looking at lipid nanoparticles for various conditions and being able to pivot science because of the capacity, because of the international work, being able to pivot in the face of this pandemic in a most amazing way. And one of the things, so I had the fortune of being in Rwanda just last week and really seeing a nation that is preparing itself for the future. It already had, as we said, some good examples of areas where it could respond quickly with technology to the pandemic, but also areas like Canada, as we said, manufacturing you didn't have, and Canada actually, in fact, didn't have, hadn't invested enough in its manufacturing, which posed quite a problem early on. So where do you see the positioning of science and its focus in Rwanda for sort of the future health systems, vaccine preparedness, pandemics? In terms of the science, there's basic science, as we just saw from Dr. Peter Cullis, that is necessary component to even get where we are today. There's also the epidemiology, the application. I met with the director of the African Center for Excellence in data science at the University of Rwanda, doing amazing work in AI and moving forward partnerships. So I'm wondering if you can comment on where you think Rwanda's scientific priorities might be going forward.
So thank you very much, Rich. I think you pointed out to an important element that we've actually seen much better during these COVID times on how the importance of science, especially on health science, is a priority for even other sectors, and it shouldn't be something to do when other things like infrastructure, road construction is completed. So that is how the actually me being in health sector, we've seen an of course, a challenging time, an opportunity to explain to our leadership, to our ministries of finance, the importance of investing now into research, into building a laboratory that does these diagnostics, that does basic science. And we acquired funding to build to upgrade our lab. We've been able to have an entire division on research recently approved by the government that we soon have been already starting to bring this data science research and innovation that we didn't have before COVID. So beyond the country level and the interest that now is being seen in Africa, in many countries, in many sectors, we see also an opportunity to work more with other researchers, innovators on our continent and beyond. There's a time we were saying Africa is contributing only 1% to the clinical trials being conducted worldwide. And today we see the importance of doing actually more clinical trials here, more diagnostics, more disease detection, more infectious disease investment than we've done before, because this virus do not respect any scientific border, being in Europe, in America, in Canada, or in Africa. So I see really an opportunity to continue working together. I'm glad you came to Rwanda last week, and we had exchanged a bit on this. We see a lot of universities coming here, researchers, and funding organizations, research funding organizations also. We should be coming in more and more, bringing us all together as scientists, as global health experts, so that we get ready and united against what the virus seems to be more united against us. So that is what I can say. But I'm glad that this meeting, this session has been also alluding to that scientific transparency and innovation has helped, but we still have challenges. For example, when a country brings in this new science, then getting somehow a victim of its own transparency, like has been said for the case of South Africa. But that should not limit us doing the right things and being transparent in research and what we see. And that should not be only for countries that have higher GDP. It should be something that we should be doing where the most diseases and most burden is. And I believe that this is why we're investing a lot into research in Rwanda and in Africa now. I hope it will continue. Thank you.
So, Catherine, I have one last question. I hope you don't mind. So, Simon, has your country also pivoted toward oral therapeutics as an alternative for treating or dealing with COVID-19? We know about Merck and Pfizer and a number of biotech companies that are working on oral therapeutics to deal with COVID-19. Is there any kind of movement in your country for that as an alternative or a complement to vaccine development and rollout?
Thank you very much. Actually, for COVID management, we've used different types of treatments, mostly monoclonal antibodies that we used since last year. We've treated more than 3,000 people using combination of monoclonal antibodies. So for this new oral therapeutics, we haven't yet started, but we're in contact with the manufacturers. We're very much interested into these two options, the Molnupiravir and also the Pfizer pill. So these options we are looking forward to as addition to what vaccine may help. But yes, we are very much interested and we look forward to these options as well.
Great, thank you so much.
You're welcome.
Excellent. Well, thank you again, Dr. Nsanzimana, on behalf of all the participants, audience today, and NGDI. I'd like to thank you so very much for joining us all the way from Rwanda.
Great, thank you. It was a pleasure joining you.
All right, thank you very much, Sabin, as well as Catherine, for this session. I know how early in the morning it is over there, so thank you so much for staying up late at night to present and give us your perspective on what's happening in your country. Amazing work going on in Rwanda. Our final session is going to be led by Dr. Nancy Galini. So Nancy, I'm going to turn it over to you to introduce our final speaker.
Thanks so much, Kish. So I'm Nancy Galini, and I'm a professor emeritus at the UBC Vancouver School of Economics. And I too am very proud to be a member of the advisory board for NGDI. So for our third speaker, we are so fortunate to have David Ridley, who is a renowned health economist at Duke University and the faculty director of Duke's Health Sector Management Program. Now, David's research examines innovation and pricing in healthcare, pharmaceuticals, and neglected diseases. And his very impactful research appears in top economic journals, as well as medical and scientific journals. But really importantly, David's research has had an impact far beyond academia and into the policy arena. And in particular, he was the lead author on a paper proposing the Priority Review Voucher Program, which became law in the United States. And it created a market of more than $1 billion for drug development for neglected diseases. And more than a dozen drugs and vaccines have received priority review vouchers for infectious diseases, such as Ebola, malaria, leishmaniasis, river blindness, sleeping sickness, and tuberculosis. So David, thank you so much for being here, and it's so great to see you again. We look forward to hearing your thoughts on encouraging vaccine development and production for the COVID pandemic and the next pandemic. Over to you.
Thank you. What a treat to be introduced by Professor Galini. She's one of the world's leading authorities on the economics of innovation. So it's an honor to be introduced by her. And it's an honor to get to talk to the team of NGDI and the broader audience here. So thanks very much for this opportunity. I want to spend 5 minutes reviewing a COVID vaccine case study, talking mostly about supply, a little bit about demand. Then I want to spend 5 minutes on an HIV case study, which I think informs how we approach COVID. And finally, I want to talk about the priority review voucher for about 5 minutes. That's what Nancy talked about. That's our paper that became law. So about 5 minutes on each, and then your questions and comments. And I am enthusiastic about questions and comments. I see this as a great opportunity to learn from a lot of experts. So you've seen plots like this. This is vaccine doses administered. And you see Canada in the dark green. A lot of Canadians, fortunately, have been vaccinated. More than in the US, in part due to vaccine hesitancy in the US. You can see that the continent of Africa is rather light colored. So that's vaccine doses administered. Here is vaccine doses secured. So commitments to get these vaccine doses. You see Canada is quite dark in this plot. 4 vaccine doses secured per person in Canada. Contrast that with South Africa of about 1, and other countries in sub-Saharan Africa similarly at numbers lower than in countries like the US and Canada. So there's 8 billion of us on Earth, 21 billion doses secured globally. COVAX, you may be familiar with COVAX. It's the WHO, Gavi, CEPI team. They have secured 6 billion doses, but they've only shipped about 500 million doses so far. So COVAX behind others, I want to talk quite a lot about COVAX today because I think it's a fabulous effort. However, COVAX behind many richer countries because COVAX trying to get money when richer countries were trying to get vaccines. So COVAX started at a disadvantage in negotiations with vaccine manufacturers because they were trying to get money and then doing the negotiation, whereas many richer countries had a pot of money. More on COVAX in a moment. A great effort, but still only about 500 million doses shipped so far, with the goal of about 1 billion by the end of the year. Many of the donations coming from G7 countries. You see Canada on this list. In part, these are sort of leftover vaccines due to vaccine hesitancy. But in part, there was a real effort even 15 months ago, which I'll show you next, to fund COVAX and provide vaccines around the world. This is one of my favorite figures. This is from The Economist. I'll let you stare at this for a moment. This is about 15 months ago. So this is before countries like the US and Canada had approved vaccines for emergency use. So on the left side, you see the countries, the organizations, US, European Union, COVAX. On the right side, you see the vaccine manufacturers. It's funny, Moderna's down there at the bottom. It's kind of an afterthought. There wasn't much public information about those contracts yet. Moderna had, we didn't know if the mRNA vaccines would work. At least I didn't know they'd work. Maybe Peter Cullis and the rest of you knew that they would work, but I didn't know the Moderna vaccines would work. And I didn't know that Moderna would work. Moderna had never commercialized a vaccine before. So Moderna kind of an afterthought down there. But you see the sort of, on the left side, you see the sort of commitments that were made. So 15 months ago, the Trump administration, to their credit, committed nearly $10 billion. That was an order of magnitude more than the entire European Union. So the European Union and US are similar scales, and yet the US had committed an order of magnitude more. And the European Union committed to AstraZeneca and Sanofi GSK, which made sense. Those were local companies, Sanofi French, AstraZeneca and GSK British. But again, credit to the Trump administration, they diversified their portfolio. You see AstraZeneca, you see the blue lines going from the US to AstraZeneca and Sanofi and Pfizer and Novavax and J&J and Moderna. So, and then you see COVAX below. COVAX at $750 million. It's a fine effort, but it really should have been an order of magnitude more than that. This should have been closer to, well, I'm going to talk about PEPFAR in a moment when I talk about HIV, but the US spends $7 billion a year for HIV drugs under PEPFAR. If the US had made a similar effort here of $7 billion back much earlier in 2020, that could have made an enormous difference for manufacturing capacity and access to vaccines. So the good news here is that the US put up 10 billion, European Union $1 billion, COVAX $1 billion. The bad news is it should have been much bigger, and it should have been more diversified among manufacturers. If I were placing bets a year and a half ago, I would have bet on, say, Sanofi. Sanofi is Sanofi and GSK, some of the world's leading vaccine manufacturers. I don't think I would have bet on Moderna, which had never successfully commercialized a product. And yet it's Moderna that had such success. Again, many of you may have anticipated that. I did not. So this sort of highlights the value of diversifying your portfolio. It highlights the value of making a big investment and recognizing that I think we underinvested, and I'll say more about that in a moment. So rich countries should have, a year and a half ago, committed big to high quantities to induce large capacity, to diversify the risk because some manufacturers would fail, to have supply sooner, and to have capacity to share with the world, and to share with the world much faster. Because really, these are small numbers. $1 billion is really a small number for rich countries like these. And so it's really remarkable that these numbers were so small at the time. Bob Hancock mentioned earlier that there are also some demand side issues. I've spoken mostly about supply side, and I think that's the right emphasis on the supply side. But we shouldn't ignore the demand side, because there's certainly vaccine hesitancy in this country, and the news recently has been South Africa putting a halt to vaccine deliveries because there's some vaccine hesitancy in South Africa. For most of sub-Saharan Africa, I think it's more of a supply issue than a demand issue. But we do need to be aware of the demand side. And that's why I'm glad that for previous speakers, there was so much discussion about demand, about vaccine hesitancy, and about the demand side, because that will be more and more. I think the supply side has been hugely important, but going forward, the demand side will become more and more important. Okay, I want to talk briefly for 4 or 5 minutes about HIV and try to relate some lessons that we have applied or should have applied today. So first of all, some failures with HIV/AIDS. First failure by drug makers. In the 1980s and the 1990s, drug makers defended their patents and charged high prices in lower income countries. I think drug makers were afraid that if they charged a lower price in another country, that would undermine their high prices in richer countries. That proved to be wrong. But I think in the 80s and 90s, they were afraid of that. I think they were tremendously embarrassed by all the harm they did. And I think they've learned from that. I'll talk more about that in a moment. The governments were slow to review drugs. Here in the US, the Food and Drug Administration would take more than a year to review the typical drug. HIV/AIDS activists said, people are dying here. Why are you taking more than a year to review these drugs? And finally, there's a lack of purchase commitment from governments to provide HIV/AIDS drugs around the world. Fortunately, there have been some improvements since then. So Gilead, Merck, Pfizer, other drug makers have learned from those mistakes in the 80s and 90s with regard to HIV. And now with their antiviral pills, they regularly license them to other countries, make them available for low prices. Gilead makes its hepatitis C drug Sovaldi available in India for 1% of the US price, and they do that by licensing to manufacturers in India. So a lot of companies learn from the stupid, even evil things they did in the 80s and 90s and have improved. Now, Gilead, Merck, Pfizer, I'm talking about the antiviral pills they make. We haven't seen the same effort with vaccines. Part of that is related to the technology, but there are other things we can discuss in that regard. So it's really, I think, a wonderful story with regard to pills. The vaccine story is somewhat different. Down to the next row, the US regulators being slow, including the US FDA. HIV/AIDS activists said, hurry up, FDA, people are dying here. In response, in 1992, Congress created the Priority Review Program. Now, that's different than the Priority Review Voucher, which was our idea, which became law in 2007. This is back in 1992. This is just the Priority Review, meaning the FDA would review important drugs in 6 months. I'm going to talk more about vouchers for 6-month review later, but for now, I'm just talking about 1992. Congress created a pathway in which the FDA would review drugs faster. Now, so that was 1992. FDA sped up to 6 months. What are we going to see with COVID? So the US FDA provided emergency use authorization for the COVID vaccines in a matter of weeks, in less than a month. So FDA demonstrated that they can review products. Now, admittedly, it's not an approval, it's emergency use authorization, but that they could authorize products in weeks. British did it even faster. Now, the key is that FDA is actually not actually conducting any studies of its own, it's reviewing data. So the question is, how long does it take FDA to review data, to convene a panel of experts, and to inspect a manufacturing facility? Can those be done faster than 6 months? I think so. Does FDA have the scientific capacity to review things faster? That's another matter as to whether FDA is able to hire a big enough team for this process. Okay, that's not what you're here for, but I thought I'd throw that out, that during HIV/AIDS, drug regulation changed because of HIV/AIDS. Will drug regulation and vaccine regulation change because of COVID? We'll see. And finally, lack of purchase commitment from governments. The improvement was under President George W. Bush, the US created PEPFAR. And here you see PEPFAR funding of about $6 or $7 billion a year. Now remember, COVAX 15 months ago was funded at 10% of this. So if we had made the same commitment to COVAX that we made with PEPFAR, then we could have done a lot more good in the world. And if other rich countries had done similarly, we would have had a lot of resources for COVAX early on, made big commitments to manufacturers, and had greater capacity earlier. And you see beneficiaries of the HIV/AIDS drugs under PEPFAR around the world. Okay, final 5 minutes, quick case study on the Priority Review Voucher. So we wrote this paper, published in Health Affairs. Fortunately, the editors of Health Affairs had a press conference at the National Press Club. I presented the paper at the National Press Club. A reporter came up to me afterward and said, I know a senator that would like this. The senator did like this. Fortunately, there was already going to be an FDA bill in 2007. It became part of that FDA bill. So it became law in 2007. So under the Priority Review Voucher Program, if you develop a drug or a vaccine for, say, an infectious disease, you get a voucher for priority review of a different drug. Now remember, Congress created priority review in 1992. That's a 6-month review. But what I'm talking about is a priority review voucher that became law 15 years later. It's a ticket, a golden ticket for a faster review of a different drug. So for every voucher, there's 2 drugs involved. For example, a drug for Zika or Ebola or river blindness or leishmaniasis. That drug gets fast review because it's important. but you get a bonus fast review for something else and you can sell that right. So vouchers have been used to speed drugs for cholesterol. They've been used to speed diabetes drugs. They've been used for HIV/AIDS combination therapies. So you win it for the neglected disease drug that has no profitability. That goes to market. That gets reviewed fast because it's important, but you get a bonus fast review for something else. So that's the priority review voucher program. There have been, I count 53 vouchers as of this month awarded. Complicating matters. So was that in 2012, Congress extended the priority review voucher program to rare pediatric diseases. So the good news is, and then in 2016 to medical countermeasures. So like bioterror and anthrax and diseases like that. So the good news is there's been a lot of vouchers awarded. The bad news is, as an economist, I can tell you supply and demand matters, and the prices come down a lot. So the initial price of vouchers was initially kind of low because people were saying, what the heck is this certificate? Is it really good for anything? Does it really work? Prices of vouchers got up over $300 million. And then with the big supply of vouchers, they're about 100 million. So $100 million is helpful for a drug developer. I mentioned river blindness here. There's a not-for-profit in Australia called Medicines Development for Global Health. They got money from investors with the hope of winning a voucher. And they did win a voucher, and then they were able to compensate their investors and have some money left over for access. Although that not-for-profit said they were going to give away their drug for free when the voucher price was around 200 million. The voucher price of around 100 million, they're going to sell it at cost. So we've got a great new drug for river blindness, but with the lower voucher price, fewer resources for that not-for-profit. In August, my colleague Hannah Kettler, who's now at Gavi, and Pranav Ganapathy. Pranav was a student here at Duke. We wrote a paper reviewing the priority review voucher program, trying to draw some lessons for motivating drugs and vaccines for infectious diseases now and in the future. A few notes from that paper. We saw a lot of success with combining push and pull funding. So push funding is upfront money for clinical trials. Pull funding is an award conditional on success. So we saw a lot of examples where drugs and vaccines benefited from both. Say, with one malaria drug, the Gates Foundation backed Medicine for Malaria venture, gave them some money up front, pushed them, and they pulled them through with the priority review voucher. A lot of the priority review voucher winning companies are companies I had never heard of before, like Medicines Development for Global Health, this not-for-profit by Mark Sullivan down in Australia. I'd say, some of the companies I've heard of, Merck has received vouchers, and GSK and Novartis, those are companies I've heard of, but most of the voucher winning companies I'd never heard of. And I think that's important when we think about how to induce product development. The great thing about prizes is you describe the product you want, and then some scientists you've never heard of, but who are doing awesome work, have a great way of attacking that problem. And many of the companies receiving vouchers have actually been smaller companies with a lower cost base, I think. So I'm a big fan of putting a prize out there. Hey, somebody developed this. I'm not going to pick the winner right away. I'm going to see who comes out. Now, it's not enough just to get innovation. You also want access. So you win a priority review voucher if you get FDA approval for this product. Unfortunately, for example, one leishmaniasis drug has not reached as many patients as we would like. The recipient of the priority review voucher was a small company, didn't have a big commitment to global health. They've been criticized for not getting the product out there. The good news is, though, that most of the voucher recipients have done a really great job of getting their products out there. Part of the reason is some of them got some upfront push funding, clinical trial funding from, say, the Gates Foundation. The Gates Foundation says, you better get this product out to people. Also, many of the companies working on these diseases, just part of their mission. So their whole mission is to get these products to people. So for the most part, access has been good, but imperfect. Okay, so what are some lessons we can take away for COVID and for the next pandemic? So you do want to use some push funding. Push funding is quite valuable. It really helps especially some of the smaller developers defray some costs, get some cash right away. But also use prizes, because you never know who might come up with a great technology and win those prizes. And finally, innovation doesn't guarantee access. Access is hard. Access is not cheap. It's not automatic. I mentioned the leishmaniasis voucher recipient that hasn't reached as many people as we would hope. COVAX has had issues with getting those products shipped and getting those jabs in arms. So don't take for granted that access is easy. I'm sure this audience doesn't, but I think an important note for other audiences. Okay, quick plug for some of my colleagues. Some of my colleagues at this place have some nice websites with some good data, and then also a plug there at the end for my webpage. My webpage is priorityreviewvoucher.org. You can learn more about the voucher program if you're interested. Okay. Questions and comments, please, because I look forward to learning from you.
Wonderful talk. Thank you so much, David. So yeah, we have 10 minutes now for Q&A, and you can raise your hand, but the hand only stays for a short time. So it'd be great if you just kind of jump in and ask questions or write something in the chat, and I'll try to get to it. I do have several questions, but if anybody from the audience wants to start, please do.
So Nancy, it's Quiche. David, great to meet you in Zoom person than by email. Great talk. The orphan drug disease designation program at the FDA, how does that play into the voucher program? And the reason why I ask is I was involved in an orphan drug designation for one of our technologies that we developed, and it helped us out. But we haven't been able to push the whole thing over the finish line. So how is that, the orphan disease program, related to the voucher, or how does that all fit in?
Yep, great question. It definitely relates, because in the United States, an orphan drug is a drug that for a disease that affects 200,000 or fewer Americans. So for these, even though malaria affects a lot of people, it's an orphan. It counts as an orphan disease in the United States because very few Americans have it, which I think is actually, it's quite helpful to have those incentives, because diseases, even though there's some incentives for malaria and TB product development, we need more because of the huge burden of these diseases. So anyway, malaria is an orphan disease, but also the rare diseases you would think of as being orphan diseases also. So I'll use my hands to do a Venn diagram. So orphan diseases are fewer than 200,000 Americans. Some of these qualify for the priority review voucher, but not all. So the ones that overlap here would be all the neglected diseases, because very few Americans have those, and some of the rare diseases, because as I mentioned in 2012, Congress extended eligibility to rare pediatric diseases. So the overlap here is rare pediatric and neglected. The orphan diseases that don't overlap are the adult orphan diseases. What do you get from under the Orphan Drug Act? You used to get 50% tax credits. Unfortunately, Congress cut that down to 25%. And you get 7 years of market exclusivity. So the Orphan Drug Act is really helpful if you are trying to repurpose an old drug that's off patent, and you don't want to make a big investment and then immediately have generic competition. So some companies and some investors have funded old drugs for new indications thanks to that market exclusivity. But I think the Orphan Drug Act has been around since the 1980s, and there's been a lot of work on orphan drugs in the United States, in part because you can charge very high prices for those because there's no competition. If it's a small enough market, there's only going to be one entrant. So some overlap, but not perfect overlap, both important programs, I think. Thank you, Keith.
No, thank you.
Great. Just a couple of comments, and then I do have some questions, but again, I wanted to come from the audience. First of all, when you mentioned that your predictions on which vaccines would be successful, it just made me think, economists, that's just a lesson. Economists are just really bad at predictions. And maybe we just need to rely more on our scientists to tell us what's going to happen with the economy. So that was, an important point you made. Another one is with the push, the push policy. Given Peter's extraordinary talk on his journey and the importance of basic research, push is usually directed towards a particular product, but we really, really do need the kinds of the funding for basic research as well. That's so important. But what does concern me is this access issue that you brought up a couple of times. And I was just wondering if the voucher program or some other program could be extended to not simply the development of a drug, but also the production and access of the drug at a reasonable price to developing countries. Do you have any thoughts on that? Or maybe there's an alternative policy that would be more efficient?
So this is just inside baseball for Nancy for just a moment. Nancy, I was just talking to a couple colleagues, Pino LaPoma and Sandro Brusco, and we're working on a paper that addresses just this very issue. So for what it's worth, I'm working with some people on applying mechanism design. Anyway, that's all I'll say about it for this audience, but I'm going to send that paper to you hopefully in a few
for sure,
weeks.
But I'm sure this audience would love to see it too.
Yes, well, good. Okay, but let me speak more broadly. So the priority review voucher awards innovation, but doesn't guarantee access. In our original paper, in our health affairs paper, in which we proposed the priority review voucher, we said, if you get the voucher, you should waive your intellectual property protections. You should waive your patent. That didn't happen, so Congress didn't adopt that. I'm not sure that really mattered anyway, because these are not profitable drugs anyway. There aren't companies just itching to sell most of these. For example, that leishmaniasis drug that won a voucher and that has had limited access. There weren't other companies clamoring to make it. So that was a flawed approach on our part. So we said waive patent protection, that didn't happen. That was probably a flawed approach. Medicines Sans Frontières, Doctors Without Borders had a nice proposal, which I very much agreed with, and it was a really low bar. I think this would not satisfy that many people in the global health community, but it was really pragmatic by MSF, and I agreed with it, but we couldn't get traction on it in Congress, and that is when you submit your application for the drug and for the voucher, you also submit your plan for access. So at least we can see you spelled out, I plan to launch the product. I plan to go through the regulatory review process in these countries. I plan to have these partners. I plan to produce this much, have this capacity. Here's where I'm going to make it. At least just have a plan. FDA would publish that plan on their website, and then we could hold drug makers accountable if they didn't meet their plan. Drug makers didn't like it for a variety of reasons. In fact, they said, access is hard. We sometimes screw up. We sometimes go down the wrong way. We don't want to be embarrassed for this. And it was generally thought, one of the excuses for why it didn't happen is we don't want to scare those folks off. So they're already sort of barely interested in working on these diseases. If we put up additional barriers, we'll scare them off. So instead, the approach people have said should be additional funding to promote access. So accept that they're just going to do the innovation, and now we need an additional pot of money for access. So those are some approaches. Require that they waive IP, require that they at least have an access plan, require that they require that they actually achieve access, or come up with a different pool of money. But I guess the fifth thing I would say, I guess, is trust that many of them actually have pretty good intentions. So, the recipient of a voucher for Chagas was a not-for-profit foundation in partnership with DNDi. Their whole mission is to serve people and get these products out there. So for at least some of the products, I think they are reaching people in part because of the mission, but there are certainly examples that are not.
Okay, great. Thanks so much. We do have a question here in the chat. Let's see. How does a group like from Liz Hancock, how does a group like Gilead or the regulators stop wealthy countries jumping onto the low price in India, in quotes, type bandwagon?
Yeah, it's a great question. This is exactly what drug makers were worried about in the 80s and 90s, and it hasn't happened. So why not? So I don't know the logistics really well, but I think if you get the hepatitis C, the Sovaldi drug in India, you have to demonstrate that you are a resident. So that's trying to cut back on some of the medical tourism. I think there's still a little bit of medical tourism, but there's not much medical tourism for these. So you have to prove that you're a resident of the country. But one thing that really helps is in rich countries, these drugs are covered by insurance and people have relatively low copays for them anyway. So as a patient, I'm getting cured of hepatitis C for some copays. And I don't have a strong incentive to try to go to another country. Pharmacists in the United States cannot re-import, say, drugs from Canada or drugs from India. So it's illegal. It's legal for you to go from the US to Canada and fill your prescription, assuming you see a doctor. It's illegal for you to go from the US to Canada, fill up a truck and bring it back. So first of all, it's illegal to move the product. That helps prevent the parallel trade. And also, insured people are getting it through insurance with relatively low copay, especially say in Europe. And so there's really not much incentive for them to shop around. So I'm absolutely delighted. As economists, we like differential pricing. We think it's efficient and fair for richer people to pay more and lower income people to pay less. So I'm delighted that this doesn't just work in theory, it's actually been working in practice for a long time.
Great, thanks. Now, according to Keish, there was a hand up, but I no longer see it. Could the person just jump in? No? Any other questions?
Yeah.
Oh, great. Thank you.
Sorry, I'm Paul. I'm from Kenya, but I'm currently in UBC. So I have some maybe few questions. Yeah, and I will target maybe two people here, David and Richard, because Richard maybe has gone to Rwanda, and I think he has also visited Kenya because I come from Kenya. So according to conversation I've had from David, there is more issues about like funding, there is collaboration. And earlier, Peter talked about the basic research. With respect to the current COVID scenario, you maybe like they started somehow from China. Maybe I don't want to go to those issues. Like now, when we talk about collaboration, funding, and basic research, you may find developing countries, maybe, I don't want to say that something is going to come from there. Maybe another disease is going to come from like a developing country, maybe from Africa, who don't have the capability of detecting. So like COVID, at least it was somehow detected somehow first from China, and then it was checked in other countries. Now, as how or where do we put these developing countries when we are talking about these collaborations, these vouchers, this funding, and what have you? Because even future diseases come from there, which are so bad than even the COVID. It may take like a long time to detect and then even prevent and then come up with the vaccines. So where and how do we put these considerations with respect to the developing countries?
I'll take a first stab at it and then I'll defer to Rich. So it's a great question about detection capabilities as diseases arise. And South Africa has really been impressive in detecting variants faster than other higher income countries. And I think your friends in Kenya will be not far behind. I was in Nairobi, I guess summer of 2019, I think. And I met with entrepreneur after entrepreneur. Your home country is incredibly entrepreneurial, I think, especially in healthcare. And so I would anticipate rapid advances in these detection capabilities in places like your home. But I defer to Rich's expertise.
I'll just comment briefly as well, because I think, we saw, I don't know if Dr. Seben was able to stay on from Rwanda, but we're seeing definitely increasing capacity in diagnostics as well as therapeutics and vaccines for sure. As they mentioned, my experience flying through both Kenya and Rwanda in the last few weeks was that, I got a PCR test I could get back in 3 hours at the airport in Nairobi, and I couldn't hope for that here. So that, and as you said, those are private companies and labs popping up, but meeting demand and response. And there is a big entrepreneurial spirit. I know Kenya, Rwanda, other places are investing a lot in health sector innovation, but and I think the world of precision health and what we've seen emerging, going back to Peter Cullis's talk in the basic science, but also in delivery and health systems, in some ways, I just see there's some advantage. They have such more of a clean slate to work with in terms of legacy technologies, legacy systems. Sometimes you see these technologies being adopted faster than anywhere. A good example in non-health was Kenya was the first country on earth to have mobile phone payment system through Safaricom M-Pesa. And that really now we get used to things like Interact transfers here, but they were way ahead back in, you can buy a mango with your cell phone text. And you can pay your staff and you can pay your lab staff. So all I'll just say is that I think, these collaborations, discussions going forward, the diagnostic side and surveillance side, it's good to see some of these investments happening, but others can probably speak to it as well. Thanks.
Well, I think that was a wonderful question to end this session on. And if you have any other questions, you can direct them to David or to me, and I can send them to David. But on behalf of the participants again and NGDI, I want to thank you, David, for your really enlightening talk, and looking forward to talking to you more about this in the future. So everyone, please clap your hands or show up your little hand.
Actually, I noticed that David Patrick still has his hand up. So David, did you want to, I didn't want to supersede Nancy there, but David, did you want to ask a quick question? Sorry.
I sure did. I'm just sort of thinking about bringing it back to the local again. Here we are sitting in this little corner of North America, and a number of us have some moral distress about the maldistribution of the vaccines. And we're saying, well, Omicron was inevitable payback and that sort of thing. So the question really is, what are the array of things that we can do that make a difference? We lobby our national governments to play bigger role. I'm not sure if we defined a role for state or provincial governments within a federated system, but they have capacity as well. And I'm wondering in particular, David, whether we've fully factored in the potential of philanthropy, whether that's already there or whether there's a way to grow that, because I think this emotion of wanting to change things globally is pretty widespread at this point, and UNICEF and other players are quite good at this. So is that all factored in with the Gavi contribution to your early diagram, or do we have a long way to run with philanthropy if governments can't do it all?
That's a fabulous question, David, and I'm embarrassed that I didn't talk more about that. So I think you're absolutely right that foundations can play a big role here, and but also the government can. And I think our government's dramatically underinvested. So going forward, we should really push our governments to invest an order of magnitude more and earlier. But we should make those philanthropic gifts. I'm really glad you made that point, David. I agree with you.
Well, sorry, David, for not seeing your hand. I was looking at the other little hand that disappears after a while, but I now see it's quite subtle there. But Kish, thank you for noticing that. And you also gave me a few more minutes. According to my clock, it was 10 minutes up, but if there are any other questions, please, I think you should just jump in. Any other questions for David or general comments on any of the talks for that matter?
Okay.
Okay, well, that was a wonderful question, David, to end David's talk on. So thank you for that. And again, once more, let us thank David for his great talk and for all your questions. Wonderful.
Okay, great. So to close out the conference, Bob's clapping. Okay, good. That's clap. The clap versus the hands raising looks similar. So at least on my screen. So to close the conference, I'm going to ask Rich to do some final conclusions, and then I have a comment at the very end. So Rich, over to you. We've got a couple of minutes.
Thank you. Thank you, everybody. This has been a tremendous. I'm going to try sharing again there. Just want to make sure I say thank you. Thank you to everyone. And I don't know if my screen's up now, but Fanan Fatah and Samia El-Jouidi from our lab has really helped put a lot of the technical parts of this together, and I just really want to thank them for helping us pull this off. We've had tremendous speakers from basic science here through UBC that's had a long path of contributing to the underlying basic science that even makes these things like vaccines possible to begin with. We've had some words from Dr. Saban Nsanzimana about Rwanda, and he apologized. He had difficulties getting his slides up, so hopefully I tried to share a few visuals, but I think he's very engaging and easy to follow with his talk. Their leadership in an ability to embrace technology, be a data-driven approach to the pandemic has really paid off, but there's gaps, and it's so warming to see that even though we're so frustrated, as David Patrick said about vaccine inequality, we're supposed to be all in this global together, and the best way to reduce new variants is to reduce the global burden of replicating virus. We can't simply just be happy with 90% or 95% vaccine coverage in Canada. If it's circulating elsewhere, then that's going to come back, as we might be seeing now. And the fact that Rwanda is already, and other, Senegal, South Africa, making investments in vaccine manufacturing for the future, probably in the science as well, not just in the manufacturing, but probably all the way through the system to underlying science and policy has been tremendous. So I've really enjoyed the talk. I hope you guys have too. Thanks, Kish. Maybe you want to do a couple of closing words on NGDI as well. But do get involved with NGDI, because I think we've gone a bit quiet in the last while because we've been through the years searching through funding. I think we've got Kish back with a lot of energy and networks, and we may be able to get this push, but we really require, I think, at all of our institutions, a platform for sharing these voices, sharing these ideas, sharing advocacy for science. So so stay tuned for us, and maybe a few closing words from Kish or any of the other board members, if you wish.
Yeah, thank you very much, Rich. And I do want to thank you and your team. Samina and Fanan did an outstanding job. It's always difficult to do these things remotely. So thank you, everybody. Thank you to the board members. They've been very supportive and played an important role as we move forward. And to Rich's point, NGDI was started as a dream, as a mission, and Bob has been fantastic in helping us getting it off the ground. Bob Hancock, who I'm speaking to about, and we really had a great amount of momentum. Rich has done a fantastic job in my absence when I decided to be a dean and go to the dark side for 5 years in academia. But we are rebranding. We are working on a new website. We've got a new active Twitter feed. We are fundraising, and I think we are excited for some new things to happen in 2022. And this conference, which I want to give Rich some great credit for, because it was really part of Rich's concept in his mind that we should have a conference like this, is only the beginning of the kinds of things we're planning to do for NGDI in the next couple of years. So we want you to be engaged. To our students who are online, and to students, please spread the word. Our students, our undergraduate students and our graduate students and our postdocs and trainees are outstanding. Their passion for what's happening in our world is amazing, and they're really the future of what's happening with things like neglected global diseases. And I would last state that I think the pandemic, if nothing else, has reawakened our concerns around these kinds of diseases. The economics, as David talked about, the on-the-ground work that needs to be done that Sabine talked about, and of course the fundamental science that we all had to pivot to. And Peter showed a great example of that, of what he had to do to pivot his research coming from a totally different background in biophysics and physics to what he's done with the COVID vaccine. So stay tuned. Please be engaged. Email Rich or I or any of the advisory board members, and please get involved. And thank you very much. And it's always great to be done early. So we're finished 18 minutes early, or 12 minutes early. So thank you, everybody, and have a great, wonderful rest of your day, and enjoy the holiday season, and please be safe. So thank you, everyone.